Welcome back. In this video, we're going to do a pretty extensive study on arsenic-containing compounds. Okay, arsenic, you should know up front, is an extremely toxic atom. Okay, it doesn't matter whether it's arsenic bound to a bunch of hydrogens, arsenic bound to a bunch of oxygens. Arsenic is toxic. And human cells, mammalian cells, bird cells, whatever it is, they do not tolerate arsenic at all. In fact, arsenic is toxic, it's carcinogenic, it can induce necrosis, it can induce apoptosis. If the cell doesn't undergo apoptosis, you'll get cancerous cells. It's just overall not a good thing. What we're going to bring out in this video is arsenic can uncouple glycolysis to where you get no net ATP. It also has the capacity to uncouple the TCA cycle. Not through the same mechanism, but what it ends up doing is it allosterically inhibits the pyruvate dehydrogenase complex, which is absolutely detrimental to the cell because it basically decreases the overall ATP production to basically nothing. And if the cell can't get energy, it undergoes apoptosis. Okay, there's all sorts of other, I wouldn't say minor health risks that are associated with arsenic, but they are a factor that we'll look at later in this video. But before we really get into the function of arsenic, we have to really understand its structure. Okay, and what I've done here, shown right here, is I have on the left one of the resonance structures of arsenic. Okay, and specifically this is an oxygen arsenic compound, and this is arsenate. Arsenate is an extremely common arsenic-containing compound. One thing you should be aware of with a compound like this is it's the most oxidized form of arsenate. Arsenic itself, if you look at the periodic table, it's a metalloid, okay? But in compounds like this, you can essentially treat it as a non-metal. Okay, so here it's behaving very similarly to a non-metal, okay? And actually what we can say is that arsenic's valence here is 5. Okay, so arsenic here has a valence of 5, and so it's able to basically have 5 bonds. It has 4 sigma bonds, and it has 1 pi bond, so its valence is 5. But this form of arsenic, this arsenate molecule, is one of 4 resonance structures. Okay, so what I can do here is let's say I take this lone pair. Well, I can move these electrons around and kick those pi electrons in there to form a new pi bond, and I get this resonance structure. And you can imagine what I'm going to do is I'm going to do the same thing to all these other guys. Okay, so then I can take this one, kick it in here to form the pi bond, these kick out onto the oxygen. And so what I'm basically showing is I'm showing the many resonance forms of arsenic. But what I can really do is I can depict a resonance hybrid in which the electron density, the pi electron density, is distributed about all these sigma bonds to each oxygen. And I tried to denote that here with these dashed blue lines. Okay, so the electron density for the pi system is distributed throughout all of the bonds in the arsenate molecule. Okay, but what's of really critical importance here is that the arsenate has these partial charges on it which make it soluble in water. Okay, so when this thing's traveling in the blood, it doesn't do so bound to proteins, although it can bind to intracellular proteins. In fact, as we'll find, it can actually bind to a lot of stuff. But it doesn't have the requirement of that. It can actually float free in solution, but it develops these partial charges. So you would imagine that arsenic is not very electronegative, oxygen is, so in each case you're going to have this dipole moment that's fairly decently sized that's going to point towards the oxygen. And you could draw that for all of these, but what you're going to end up getting are partial negative charges on all the oxygen atoms. Okay? And so that leaves a partial positive charge on the arsenic. So this thing's polar. That means it can basically float free in solution, whether it be in the blood or the inside of the cell, because this guy actually can make it inside the cell, which is where it exerts its most detrimental effects. Okay? Now, this right here, this is not a resonance hybrid. This is basically just the common structure of arsenate. but I tried to draw it using three-dimensionality. Okay, so basically these bonds right here, these I'll do in yellow, these bonds right here are in the plane of your screen, and then of course these two bonds to the oxygens out here, the one right here, this one is coming out at you, this one's going back. And because every one of these bonds is equal, okay, in this form of arsenic, that makes this angle theta equal to approximately 109.5 degrees. And that's going to be the same for all of these. So you could also put an angle theta here, you could put an angle theta there, you could even put an angle theta there. They're all going to be approximately 109.5 degrees, which tells you that the overall geometry of arsenate is going to be tetrahedral. Okay, so this is a tetrahedral molecule, and it might be a little bit unintuitive to you that this is a tetrahedral molecule, because most of the tetrahedral molecules you're used to seeing have 4 bonds, but this has 5. But remember that arsenic, if you look at the periodic table, it's in the 4th period. Okay, when you're in the 2nd period, which is normally what you deal with in organic, you've got carbon, nitrogen, oxygen, and so forth. Those things really do not violate the octet rule in terms of getting more than 4 bonds. They're confined to really having 4 bonds or less. But when you get below that, when you get into the phosphorus and sulfur and arsenic and selenium in the 3rd and 4th periods respectively, they can violate the octet rule. And so they can have 5 bonds. In fact, sulfur and selenium can actually have 6 bonds. And ordinarily what they'll have, in most cases, sulfur and selenium will get 4 sigma bonds and 2 pi bonds, whereas things in the 5th period, or excuse me, in the 5th group, like arsenic and phosphorus, they'll tend to get 4 sigma bonds and 1 pi bond. So that begs the question, if arsenic is truly in group 5, it should behave very similarly to phosphorus, and in fact it does, and that's part of the way it exerts such detrimental effect. I actually have, we'll do this later, I actually have a periodic table shown here. Okay? Notice that if you look at group 5 here, you have nitrogen, which can't violate the octet rule, but below that are phosphorus and arsenic, and they can. Okay, so they can have multiple bonds, particularly more than 4. They can have 5 bonds. Usually what they'll have is, in their most oxidized form, they'll have 4 sigma bonds and 1 pi bond. And so if we come back up here and look at arsenate, let's actually draw phosphate. Because if you're in biochemistry, you're probably used to seeing phosphates. So phosphate will ordinarily be drawn with the double bond to the oxygen going up like this. You'll have one oxygen coming like this, and then you'll have one oxygen atom that comes out at you like this, and then another one that goes backwards. So this right here, this is inorganic phosphate. And it's going to behave very similarly to arsenic, and that's part of the way it exerts such a detrimental effect to the cell. Well, you could also make the argument that this angle is the same as there, and in fact it is. It's about 109.5 degrees. But humans and mammals in general have the capacity to tolerate phosphate. Of course, it's in DNA. It's the intracellular buffer, so humans certainly have the capacity to tolerate phosphate, but we don't have the ability to tolerate arsenic. Let's actually look at the first reason as to why that is. So hopefully at this point you have a little bit of a grasp about the structure of arsenic. It's a highly resonance-containing molecule, has tetrahedral geometry in its most oxidized form. In its most oxidized form, the arsenic atom has a valence of 5, okay? And it looks very similar to phosphate. It looks very similar to phosphate. In fact, enzymatically, it behaves almost identically. Okay, so what I'm going to do now is I'm going to show you steps 6 and 7 of glycolysis. So when you start the payoff phase in glycolysis, you're of course going to have, per glycolytic run, you're going to have 2 glyceraldehyde 3-phosphate molecules. I've only drawn one here for simplicity, but glyceraldehyde 3-phosphate is going to react with G3P dehydrogenase, which is going to give you this molecule, which is 1,3-bisphosphoglycerate. Okay, this is a really critical molecule because it will then go and react with phosphoglycerate kinase, which gives you this guy right here, your first ATP that is created by glycolysis. In fact, because there's two G3Ps, okay, you end up getting two ATPs, which basically accounts for the two that were lost earlier in the preparatory phase. And of course, that gives you 3-phosphoglycerate. Okay, but to understand arsenate's detrimental effect to the cell, you actually have to look at the mechanism of this guy right here, glyceraldehyde 3-phosphate dehydrogenase. Let's actually do that. Okay, so what you end up having in the active site is, I'm going to do the mechanistic steps in yellow. You have a base here in the active site, and then you have this cysteine here. Well, the base is going to deprotonate the cysteine and induce nucleophilic attack on the carbonyl that's carbon 1 of glyceraldehyde 3-phosphate, and that generates a tetrahedral intermediate. Okay, and that tetrahedral intermediate is shown right here. When the tetrahedral intermediate collapses back down to form the carbonyl, it's going to generate your first NADH. So you have to have a leaving group. Carbons generally aren't your leaving group, so your best case is either the cysteine or the hydrogen, which in this case is a hydride. Well, according to the mechanism of the enzyme, the hydride leaves, and it attacks this top position on the NAD ring, and that generates your first NADH, which is shown right here. So now what you have is you have this thioester linkage between what was glyceraldehyde 3-phosphate and the cysteine residue of the protein. Now, ordinarily what would happen in this step of the mechanism is a phosphate would come into the active site. Okay, phosphate would come in, and let me do this in orange. It would attack this carbon right there. Okay, it would just induce another nucleophilic acyl substitution, and what you would theoretically get is this guy right there. So the phosphate that comes into the active site would be this one right there. But that is not what happens when you have arsenate present, because what arsenate does is it actively competes with the phosphate. So if you have lots of arsenate present and this enzyme gets a hold of it, the enzyme cannot distinguish the difference between the arsenic in the center and the phosphorus. Okay, so what that means is that glyceraldehyde 3-phosphate dehydrogenase cannot discriminate between inorganic phosphate and inorganic arsenate. So what ends up happening is, back to yellow, the arsenate will do a nucleophilic attack right here, okay? Again, you generate another tetrahedral intermediate as shown right here. So what ends up happening is the tetrahedral intermediate collapses, and that expels the leaving group, which is the thiolate of cysteine, as it reabstracts the proton from the base, and you regenerate the base, and you end up with this guy right here. This is called 1-arseno-3-phosphoglycerate. And that's distinguishing it from 1,3-bisphosphoglycerate, which is the one that was shown right here. So this guy right here, this one is 1,3-bisphosphoglycerate, or 1,3-BPG. This is a different compound. This is 1-arseno-3-phosphoglycerate. Okay, but here's the problem. The next enzyme won't get a chance to deal with this because this guy spontaneously hydrolyzes. Okay, and even if the other enzyme was able to use this, this is an arsenic. So you wouldn't generate ATP in the next step, because this group right here, if it was a phosphate, this is what would get transferred to the ADP. So even if the arsenic was still there, it still wouldn't generate ATP, but it wouldn't matter in any case, because this is a really unstable compound. So this is unstable, and it spontaneously hydrolyzes in solution. And when I say spontaneous, I'm using that term very loosely. Of course, it has a negative delta G, but this reaction is non-enzymatic. So water comes in, hydrolyzes off the arsenate, and you end up with 3-phosphoglycerate, so 3PG. But notice that because we didn't get to react with phosphoglycerate kinase in this scheme if we use arsenate, we end up losing out on generation of 2 ATPs, and that leaves the only possible production of them in this pathway to pyruvate kinase. Okay, so because we lose out on 2 ATP, this is termed an uncoupling of glycolysis. So this is important. This is an uncoupling reaction. Okay, and the reason it's uncoupling is because we use a different substrate, excuse me, which is arsenate, and we end up losing out on the capacity to produce the ATP. So basically, even if we get the 2 ATP from pyruvate kinase, because we're going to end up having 2 of these guys, 2,3-phosphoglycerates, we're still going to get a net of 0 from glycolysis. Okay, and that leaves all the other pathways to their devices to making the ATP. Okay, but it turns out that there are other detrimental effects of arsenic-containing compounds that are not arsenate, but include it as well, like these right here. So if we scroll down, here are some other arsenic-containing compounds. We have arsenate, which we just talked about. We have arsenic trioxide, cacodylate, arsenite. Of course, it's not limited to these guys, okay? But these are just some of them, and what they can effectively do is allosterically inhibit the pyruvate dehydrogenase complex, which is what we're about to get ready to talk about. So at this point, we've talked about how arsenate, which of course is an arsenic-containing compound, uncouples glycolysis, decreases the overall energy yield, but that's not arsenic-containing compounds' only detrimental effect. You can probably see where we are right now that we're going to be talking about the pyruvate dehydrogenase complex, which is an extremely important enzyme in the production of the vast majority of energy in the cell, because as we know, when we use the pyruvate dehydrogenase complex to convert pyruvate into acetyl-CoA, the acetyl-CoA would go into the TCA cycle, and that leads to a ton of energy production that will fuel the pumping of protons by the respiratory chain, which fuels ATP synthase for the production of ATP. And what I'll go ahead and say right now, we'll go into this a little bit later as to the mechanisms of this, but just know that there are many, many arsenic-containing compounds. We already discussed arsenate and the properties of arsenate, but here's just some arsenic-containing compounds that are of extreme importance. Arsenate we've already talked about here, that's on the left. Arsenic will also form stable oxides. In this particular oxide, this is arsenic trioxide. Sometimes you may hear this referred to in the literature as diarsenic trioxide. That's, of course, because in here there's 2 arsenic atoms per unit of this complex. Arsenic trioxide is not a metabolite of arsenate in any way. Arsenic trioxide is something that you would inhale or you'd consume accidentally, perhaps at some job site, or if you're working in a chemistry lab of some sort. This is not a metabolite of any sort. This is something that would be accidentally input into your body in some way. Cacodylate can be made synthetically, and you can get it that way, but cacodylate is also a metabolite of arsenate, and we'll soon see that in the next video when we talk about the metabolism of arsenate. So this is cacodylate. And this is arsenite, the reduced form of arsenate. So arsenate, we mentioned this already, but arsenate right here has a valence of 5. Okay, if you were treating arsenic as a transition metal where you would have to put the charge on it, or the oxidation state, in Roman numerals, you can do that. Sometimes you'll see it with this 5 written here. But what you'll also see here is for arsenite, you'll see this was a 3 because this has a valence of 3. Arsenite has an arsenic atom with a valence of 3. Okay, but all of these compounds are related in the sense that they all have arsenic in them, and they're all going to have very similar effects. Some of the effects might differ in their affinity for the pyruvate dehydrogenase complex, but they're all going to have the same general effect, and humans, mammals, birds, whatever, really do not tolerate arsenic. Before we go into what it does to the pyruvate dehydrogenase complex, I just want to go over some of the very general functions of arsenic, and I really shouldn't say function, I should really say detriment. And I'm just going to go over these really quickly, but these are some important things right here. Okay, arsenic inactivates nitric oxide synthase. Nitric oxide synthase is an inducible enzyme. So nitric oxide synthase is the enzyme that takes a molecule of arginine, electrons from NADPH, and molecular oxygen and converts it into nitric oxide NO, which is a free radical, and then also L-citrulline. Okay? And the nitric oxide is used for things like vasodilation, so dilation of blood vessels, allowing oxygen to get to different areas. As a result of this, it can cause things like oxidative stress, which leads to production of free radicals. So a lot of times what happens is you get increased hydrogen peroxide concentration. And so you'll have to have enzymes to deal with that, but the oxidative stress that's caused by arsenic has to do with the fact that it increases the concentration of hydrogen peroxide. Okay? It also leads to platelet aggregation and fibrinolysis. Okay? It can induce arrhythmia. And a lot of these inflammatory molecules like interleukin-1, tumor necrosis factor alpha, and some other things, these get upregulated. These are, of course, peptides, and they get upregulated when you have a lot of arsenic, which tells you that arsenic induces an inflammatory response. So arsenic intrinsically is inflammatory. It's inflammatory, which is a little bit surprising because it's just a metalloid, but it turns out that if you're getting TNF-alpha, interleukin-1, and so forth, it is inflammatory, and arsenic can cause necrosis due to oxidative damage. And when we say oxidative damage, we of course mean damage from things like hydroxyl radicals, hydrogen peroxide, and so forth. Okay, so all this stuff, this is all caused by arsenic-containing compounds. Now, what I want to mention, and I'll circle this in orange, it causes necrosis, but there are some mechanisms that I don't have written here by which arsenic can cause things like apoptosis. Okay, some other functions I want to talk about very briefly before we get into the really important physiological properties of arsenic is that arsenic has the property to bind to two very important proteins. One of these is actin. Actin is a protein, a filament that's found, say, in muscle proteins. It's, of course, found many, many other places. But a really important one for the purpose of apoptosis is that you have tubulin, and arsenic is able to bind to tubulin. And tubulin is, of course, the monomer protein that makes up microtubules. Okay? And microtubules are important for things like cell division. So basically, whenever you have mitosis, cell division, you ultimately have to use microtubules. We know that microtubules are used for lining up the chromosomes on the metaphase plate. So if you were to somehow damage the microtubules, you're going to affect mitosis. And as a result of this, the fact that arsenic is able to bind to and damage microtubules by binding to tubulin, it's able to induce things like aneuploidy, polyploidy, really detrimental things, and ultimately what it's going to lead to is apoptosis. Okay? For other reasons as well, we'll go back and look at a very important other reason as to how it causes apoptosis, but it causes aneuploidy, polyploidy, all sorts of mutation-like things. But also what it's able to do is it's able to affect DNA methylation. So for some unknown mechanism, arsenic's able to affect DNA methylation, which of course messes with the gene expression in cells, and it can also lead to cell proliferation, which is of course characteristic of cancer cells. So not only can arsenic induce apoptosis, but if the cell division is left unchecked, it can ultimately induce cancers. Okay, cancer growth. And so as a result of this, we say arsenic is a carcinogen. Okay, so different arsenic-containing compounds have different abilities to do this, but in general, arsenic-containing compounds are carcinogens. And of course, they have all these detrimental effects right here. Okay, and of course, these arsenic-containing compounds that are listed here, these are just a short list of them. There's, of course, many others, and we may get to those in other videos, but I just wanted to expose you to just a few of these right here. These are some really common arsenic-containing compounds. Okay, now back to this. This molecule right here, this is pyruvate, and we know that pyruvate is a product of glycolysis, and we already know that arsenate is able to uncouple glycolysis, so you get no net ATP from that scheme. You'll still get the pyruvate, which will of course go into this enzyme, but here's the problem with arsenic-containing compounds. They are what we call allosteric inhibitors of this enzyme right here. So arsenic-containing compounds allosterically shut off the pyruvate dehydrogenase complex. And a lot of them shut it off very strongly. So that's what this X means here. This X means that they allosterically turn this enzyme off. Well, if you allosterically turn this enzyme off, you, number one, lose out on this NADH, which would ultimately fuel NADH ubiquinone oxidoreductase for the pumping of protons to power ATP synthase. Oops, that was not supposed to come up. Sorry about that. So you end up losing an NADH that would ordinarily go to fueling NADH ubiquinone oxidoreductase for the pumping of 4 protons for the powering of ATP synthase, but you also lose out on an acetyl-CoA, which would ordinarily go into the TCA cycle. You'd of course get 3 NADHs per cycle, 1 nucleoside triphosphate, either GTP or ATP, and then you'd of course get 1 totally reduced ubiquinol. All those things power the respiratory chain for the synthesis of ATP. But if you're not doing this enzymatic reaction because this enzyme is allosterically turned off, you're not getting the NADH from this reaction. You're not getting the acetyl-CoA for this reaction. So ultimately what's going to happen for the most part is the TCA cycle will in large part shut down because this enzyme is turned off by the arsenic-containing compound. And so by lack of energy, the cell will automatically undergo apoptosis. So you will not get through the TCA cycle. Instead, what will happen is apoptosis. Okay? So not only are arsenic-containing compounds able to induce things like necrosis through their interactions with actin and so forth, but through other mechanisms they're able to cause cancer, through unchecked cell proliferation, through messing with DNA methylation and so forth. But they also allosterically turn off this enzyme, so they're able to destroy the function of the TCA cycle. They uncouple glycolysis and so forth. So they're really detrimental compounds. Okay, but there's another arsenic-containing compound that behaves a little bit differently than the rest of these arsenic-containing compounds. Most of the arsenic-containing compounds that you'll come across, they're oxygen-containing. But there's another way that arsenic can behave, and it can behave very similarly to a nonmetal. Okay, so this molecule right here, this molecule that's shown here, this is called arsine. It's called arsine. And you notice that arsine looks very similarly to, say, ammonia. But what arsine is able to do, first of all, let's have some intuition. Arsine is not a normal biomolecule. Humans certainly don't synthesize it. We don't do any biosynthesis with arsenic as far as anyone can tell. All we do is degrade it. But essentially what arsine will do is it will be inhaled. Usually if you're working in some kind of chemistry lab or some setting in which you can inhale it, it'll go into the nasal mucosa, get inhaled into the lungs, end up in the blood, and you'll have to deal with it from there. But what it's able to do is it's able to attack hemoglobin. So this right here, this is basically hemoglobin. You know, you have your alpha subunit, a beta, another alpha, another beta. So this is hemoglobin. And I tried to draw the hemes in there just so you could see it. So arsine is able to attack hemoglobin, and what it's able to do is induce something called hemoglobin urea. So arsine is able to cause something called hemoglobinuria, okay? And that's basically when you have the destruction of red blood cells. Okay, so the arsine will destroy the red blood cells, causing symptoms of anemia, stuff very similar to that, because basically it's basically hemolytic anemia because you're destroying the red blood cell. You destroy the hemoglobin, and so the remains of the hemoglobin you'll see in the urine. So the way you detect hemoglobinuria is by looking in the urine and detecting the hemoglobin there. Okay, or the remains of it. Okay, so arsine is able to destroy red blood cells, or what we typically say are erythrocytes. Okay, so arsine is able to do that. So it behaves a little bit dissimilarly from the rest of the arsenic-containing compounds. As far as anyone knows, arsine really doesn't seem to do much with the pyruvate dehydrogenase complex. Its main function, or its main detriment, you could say, is attacking red blood cells. But the ones that are oxygen-containing, they tend to deal with the pyruvate dehydrogenase complex, and they'll have these other functions that are shown here. They'll be able to cause necrosis by, interactions with actin. They'll be able to cause mutation, cancer, growth, and so forth, and they're also inflammatory. Okay. In the next video, we'll talk about how you detoxify arsenic. And there's actually a few ways that you can detoxify arsenic-containing compounds. You can actually bind them with a protein called metallothionein. We'll have a whole video on metallothionein, but that's one protein that's able to pick up heavy metals. We actually have a catabolic pathway for arsenate. So arsenate's a very common molecule that contains arsenic that you could ingest, and we have a catabolic pathway for that, which we'll do in a future video. But I hope this makes sense. Arsenic-containing compounds that are oxygenated, okay, basically these are going to affect the pyruvate dehydrogenase complex, allosterically turn it off, and basically give the cell so little energy that it undergoes apoptosis, okay? But on top of that, they have all these other functions right here that lead to cell necrosis and ultimately lead to, if the cell survives, it can ultimately turn into a cancer cell, okay? Because of affecting DNA methylation and causing cell proliferation. And it can also mess with the DNA repair enzyme machinery. So if you destroy the ability of the cell to repair its DNA, it will also likely become cancerous if it doesn't already undergo apoptosis. Okay?